Okay, we're going to go ahead and get started. Welcome back, everyone. It's my absolute pleasure to introduce our next speaker, Dr. Kjiersten. Kjiersten is the Chief Informatics Officer at the Joint Genome Institute, JGI, which is a DOE national user facility. In her leadership role there, Kjiersten has been setting the strategic direction for computational biology and bioinformatics approaches. She's been involved in data science and informatics for a number of years, including her previous role at NERSC and Berkeley as data science engagement lead, and has been an advocate for bringing in user-centric design and thinking, which is something that we have talked about. So I've known Kjiersten a number of years. We've had lots of interesting and insightful conversations on research data, and looking forward to our presentation. So thank you, Kjiersten, for joining us today, and the floor is yours.
Well, thank you for that kind introduction. I thought I might start this presentation by describing a little bit about what the JGI is, and then telling you a little bit about our data management infrastructure, and then how we're transitioning into the need for more distributed infrastructure. And I'll assume, unless somebody says something, you can see my slides. So the JGI is, as Rashna pointed out, a national user facility. In the last year, we actually moved into this new fancy building called the Integrative Genomics building. Prior to that, we were located about 20 miles away from the main Berkeley Lab campus. And the crazy thing is that we've actually spent more time working remotely than we spent working in the new building because of the pandemic. So we're all pretty excited to get back to work and to be on site again, because the Berkeley Lab campus is pretty amazing. The mission of the JGI is to provide researchers with access to advanced sequencing capabilities. So we do a lot in the laboratory to be able to measure characteristics about microbial communities and plants and fungi, basically all of the techniques that are being applied to humans, which are super interesting, and there's a huge industry around that. We kind of take those and adapt them and think about how to use them to understand the environment, since that supports the mission of the Department of Energy. The user facility itself actually was initiated as part of the Human Genome Project, and I think that this is pretty cool history. So it's one of the times that you had true cross-agency collaboration to get something done. And I don't think that a lot of people understand or know that the DOE Department of Energy was really integral to the Human Genome Project succeeding. And you might step back and say, well, why would that be the case? And, you know, DOE, Manhattan Project, energy, radiation, nuclear power. And so really understanding how radiation impacts human health became a focus of the DOE and part of the Office of Science. And that was part of the reason that as sequencing the genome actually became feasible or potentially possible, that DOE was engaged from the outset. And so once the human genome was completed, the DOE had all of these capabilities to produce sequence data, and sequencing technology was really changing. It was becoming a lot easier and cheaper to be able to generate genomes, but it also required specialized infrastructure and specialized skills. So JGI actually turned into what was initially a production sequencing user facility, producing some of the first genomes for plants and algae and microbes and fungi. Some of the first metagenomes came out of the JGI. Metagenomes are kind of a synonym for thinking about microbiomes, which potentially most of you have heard about. And then over time, it has progressed to really becoming a place for integrative genome science. So maybe you've heard about the ability to generate a genome, but maybe you're less familiar with things like transcriptomes or metabolomes or metaproteomes. And so there's something called the central dogma of biology that describes how you actually go from this blueprint of the human genome to the actual proteins and enzymes that are generated that, you know, keep us alive and let our bodies do certain things. And so having the genome really isn't enough. You need to layer on additional information to truly understand what's going on. And even more than this, when you take something out of the environment and you sequence it, you're just getting a snapshot of what that organism was up to when you captured it and brought it into the laboratory. So it's becoming really, really important to be able to actually go out and take multiple samples of different environments to really understand what all of these microbes might be doing. And that's really this idea behind gene and genome function. It's all very well and good to have a set of characters that you might be able to do something with, but like if you don't understand what that gene is or what it's doing, then you don't really have true understanding of how this blueprint is enabling us to live and survive in different environments. So this is just a snapshot of what the JGI offers at this point in time. DNA and RNA sequencing. We also do DNA synthesis, which is pretty cool. We'll take constructs that a scientist would like to put in, that they found in one bacteria or one, yeast or another organism, and then we make it possible to actually put those constructs into a different host. And the reason you might do that is because some hosts are easier than others to be able to grow in a controlled laboratory environment. They might also have other desirable characteristics, like maybe not being a pathogen or something that could infect plants or humans or animals. We also are looking at and offer metabolomics to the broader community, which means that you can actually see and look at the spectra of a sample and understand what metabolites have been produced by that microbial community. So it starts to paint this more holistic picture of what's actually happening in the environment. JGI is also a leader in generating reference genomes for plants and fungi, and now we're moving into algae, as well as having a pretty robust program for looking at viruses. So the way I like to think about this, because I'm not a biologist, is being at JGI has really taught me that it's this unseen world that actually drives the health of humans, the health of the planet. And the more we can start to understand how these things all interact with one another, the more of an understanding we have about how we impact the environment or steps we can take to actually potentially improve things and work towards solutions for climate change, for example. And so viruses are they're just kind of cool. They're the next kind of piece in this puzzle of things that we can't see that have a huge impact on other microorganisms and on animals, as we've experienced in the last year. But it's not just the pathogenic viruses. There are all kinds of other small and large viruses that help these communities do different things. So it's, I don't know, anyway, pretty interesting. Another reason you might care about this is environmental genomics will enable what we've been calling the bioeconomy. At some point, it would be nice to transition off of fossil fuels. And I hope that's not a controversial statement in this group, but fossil fuels will run out. Fossil fuels are the precursors to a lot of different products that we rely on every day. You're probably wearing something that has a fossil fuel precursor that went into making it. And so just imagine if we could actually substitute those chemicals or those precursors with chemicals that are produced instead of through a fossil fuel process by microorganisms, by bacteria, or by yeast. And so that's kind of the idea behind this diagram that you see here. You can go from the DNA. Once you understand the function of the genes in those DNAs, you can start to think about building what they're calling genetic circuits, which can then be used to produce enzymes. And if these enzymes have desirable characteristics, you could then take that genetic circuit and put it into a microbe, and then that microbe would produce the enzymes that you need. That is a very gross generalization of how this process works, and I would encourage everyone to read more about it because it's really interesting and cool. And I feel like the more we understand the blueprints and genes and functions, and then there's also this relationship to CRISPR technology that's been developed recently, the faster we're going to be at being able to harness microbes to basically build greener fuels and greener precursors for different materials that we rely on. So JGI said is a user facility, and what does that mean? That means we support scientists across the globe and in conducting their research. So the scientists will go out, collect a sample, extract DNA and RNA from that sample, and then they'll send it to us for sequencing and analysis, or they'll send us samples where they'd like to run the mass spec on it. And we've got the predominant user base in North America, but you can see this distribution of users across the globe. And so these are primary users that are actually working with JGI to generate data, and eventually that data makes its way into the sort of public domain because it's a federally funded activity. And over JGI's history, there have been more than 100,000 projects that have been completed and over 2,300 publications that have come out as a result of this organization existing and supporting the broader scientific community, which is pretty impactful. And one of the other things to understand is that JGI adapts to new instruments and new technologies. So there was a time where short-read sequencing or Illumina sequencing or next generation sequencing had made things really, really inexpensive to generate really, really short pieces of DNA. So those are called reads that come off the sequencer. And they were incredibly accurate. And so what you could do is you could take all of those reads, you could stitch them together, find areas of overlap, and reconstruct a genome. So what you get out of the sequencer isn't a whole genome, you go through a process to actually put that together. Then along comes this long-read sequencing. And you might think to yourself, well, if you have highly accurate short reads and you can stitch them back together to create a whole genome, why would you be interested in long reads? And one of the things about studying other organisms in the environment and plants and fungi is that they have very complex genomes with very highly repetitive regions. And so if you've got two short pieces of a string and they repeat, you're not exactly sure which place in the genome they should go or how you should branch things. And so long reads really help you resolve these repetitive regions to let you get a more complete picture of what the genome looks like, which is important when you're tracing this back and thinking about how this all leads to function. And so this new technology for long read sequencing is really desirable for some of the more complex organisms like plants and also for metagenomes. So what a metagenome is, it's also pretty cool. You collect some soil from the environment and there's all kinds of stuff in there. There are going to be dead microbes, living microbes, fungi. You might have some animal DNA and they were pretty good at filtering that stuff out. But basically you've got within a single sample, hundreds, if not thousands of different organisms. And when you extract the DNA from that sample, you're getting DNA from everything. And so it all kind of gets jumbled up together. And if you've got things that are closely related, it might be really hard to resolve whole genomes and figure out which organisms are actually present in that metagenome or microbiome sample. So when you can shift to something like this long-read sequencing technology, it lets you filter out and actually pull out high quality whole genomes from the sample, giving you very good confidence that the organisms that you found are actually represented in that community. So this transition from short read to long read is beneficial scientifically, and also this technology for long read sequencing is improving, and we're seeing a large increase in demand for the use of this technology, though we're a little bit capacity limited because it is a bit more expensive than doing the short read sequencing. Anyway, it's pretty cool. And so this is just a slide that I kind of threw in after Susan's talk, because she mentioned this idea of data resources becoming more available, this idea of being able to support reproducibility and reuse of the data. So this is a snapshot of JGI and our FY20, just data access and data downloads. So as I was saying, users provide these unique samples. The samples become data through sequencing or other instrumentation processes, then get analyzed and then are made available through JGI's various different web portals. And so each of these portals, there's the data portal, which is everything that JGI produces. And then phycocosm is for algae, microcosm is for fungi, phytosomes for plants, and IMGM is for everything microbes and metagenomes and viruses. And so this is really how the user community and the broader scientific community interacts with JGI's data. In the last year, they downloaded millions of files and over a petabyte of data, and we've been seeing that trend increase. And so we keep asking ourselves a question of basically, how can we make the data even more accessible and easier for people to find? And I'll touch on that a little bit later. But there's an effort within the DOE Office of Science to really think about public reusable research data. And Susan mentioned Laura Biven, who's transitioned over to NIH. And this was an effort she had a heavy hand in at the DOE, and it finally got launched last month. So I've got the URL here for you all to take a look at. There are different groups within the Department of Energy that have gotten this sort of SC, Office of Science Public Reusable Research Data classification, and the JGI is one of them. And what that means is you're making sure that the data are as fair as possible, which I will also come back to in a bit. So we've had this massive increase in metagenomes and genomes that are getting produced at the JGI. Metagenomes, like I said, are these collections of genomes or complex samples that get sent to us. And this has just become a growing interest because we not only have the computational power to be able to analyze and tease apart these metagenomes, but we also have some of the laboratory techniques that make it possible to really kind of look at these communities and understand what's going on. And this, like I said, is really valuable when you're considering what a community is doing within a soil or a marine environment or the human gut. And so there's a lot of crossover with what some of the NIH researchers have been involved with, like the Human Microbiome Project. And then there are other NSF funded groups that are looking at the Earth Microbiome Project. And then the DOE has launched recently the National Microbiome Data Collaborative to look at bringing all of these data sets together. And so we're producing a lot more data that is really complex, computationally intensive to analyze and understand, but provides a wealth of really interesting biological information. And right now, JGI manages about a 10 petabyte data repository that's getting added to every time. And something I thought would be fun would be to tell you a little bit about the scale and the scope of microbes within the Earth. So I think everyone here is familiar with the prefixes on the size of data. And I think Susan also talked a bit about exabytes of data. And so if you really want to kind of blow your mind a little bit, is that it would take 10,000 yottabytes to actually take all of the unique genetic information present in microbes across the Earth and store it. And that's just looking at the differences between all of the genomes, not whole copies of every genome, because you could take one microbe and then use that as a reference and just look for the diff between any subsequent microbe. And so you're already trying to reduce the data, but it would take 10,000 yottabytes, which I think is kind of insane. And then the cost, and granted, this was back in 2011, storage has gotten cheaper, but still 1 yottabyte, $100 trillion. There was another statistic that said that you would need to have data centers covering Delaware and Rhode Island to store 1 yottabyte of data. That also changes over time, and that was more than 10 years ago. But it's still an interesting thought experiment about how we're going to deal with all of this data. And this is just the genome. And like I mentioned before, we want metabolomics information, transcriptomics information, proteomic information, and then to combine that with image data. And you're just using this to build hypotheses. Eventually, you also want to do laboratory experiments that actually validate that a gene has mapped to a particular function in a microbe. And I'll tell you, one of my favorite microbes or microbial functions is there are microbes out there that can actually secrete perfect magnets. And that's just so weird and cool. It's like something from a sci-fi novel. And if you think about that, like understanding the mechanisms that let them do that, there are other microbes that actually secrete perfect like calcium carbonate discs that could potentially be used to construct new biomaterials or bio-inspired materials moving forward. And so understanding how they do these things and what they need, what kind of environment they need to do these things, I think is really potentially impactful. But then you have a hypothesis about the gene that actually enables, or the set of genes that enables a microbe to be able to secrete magnets, and you want to test that out. And so you've got to be able to do that experimental validation so that you can verify the data for the community, and it just becomes this very complex process. So right now I would argue that biology is very heavily in this sort of cataloging stage, at least for microbes, and we're really trying to collect a lot of information so that we can start to look for patterns and really understand what's going on. And this is another slide about just what we can do now versus where we want to be. And the advances that we've had in the wet lab and with different instruments and technologies have really enabled this community to generate a ton more data than our data infrastructure is really ready for. And that's just become one of these barriers that we talk a lot about how to overcome. And this goes hand in hand. I believe there were some comments about metadata and metadata curation. Like you can generate all the bits that you want, but if you don't have the corresponding metadata or context to be able to find it later, it's not terribly helpful. And so in addition to just being able to have the capacity to do the kind of analysis that you need to do, you've got to have the corresponding like data management systems in place to help you do that. And so one of the things that we're focused on at JGI and with the National Microbiome Data Collaborative is how can we remove the barriers to data access and analysis at scale? What kind of resources are needed? And you know, we haven't had as much luck as NIH has with the cloud, but the Department of Energy does have huge computational resources available at our disposal. So we try to make creative and clever use of those. So I just want to come back to this idea of like, we have this huge, enormous growth happening in these complex data sets that are being generated. Also, metagenomic data sets are on the order of terabytes in size, just the raw data. So passing those around a lot can get to be kind of expensive. You know, back maybe it was 2013, which if you look at this curve is sort of before this massive explosive growth that we had in just sort of the number of data sets that were being generated at JGI. Data management within our organization looks something like this. And so you can kind of ignore the acronyms and just think of each box as being a group within the Joint Genome Institute that needs to share data with other groups across the organization. And then eventually all of that data needs to go into one of our outward facing science gateways or user portals so it can be accessed by our external collaborators. And so everybody was talking to everybody else, and they had different strategies for how they would keep track of data. And where it was on the file system was a very popular choice. So using the file system like a database, I think most people on this call would know that that doesn't scale terribly well. So we deployed a hierarchical data management system to deal with all of this growth we were experiencing in data and also to help us get more organized. And so the after picture looks something like this. So we have something called the JGI Archive and Metadata Organizer, or a JAMO, cute name. And you know, we actually are able to take advantage of the tape archives within the national labs to store and manage data and really only keep it on spinning disk when there's active analysis being done. Now all of the groups within JGI have a central resource to be able to query to find the location of the data to figure out if it's available for processing. And what this has done, and this is the backend infrastructure. So we have a file manager that's in SQL, but all of the metadata that describes each of these data objects that's managed by JAMO sits in a Mongo database. And there's connectivity to all of the NERSC file systems as well as to the archive. And this really enables automation as well between the different groups. So the core pipelines connect with JAMO and they provide templated metadata. And so the reason that's helpful is because everybody agrees on the same core sets of keys so that they know how to find that project information later. And once data is available for processing, the workflows are automatically triggered. And basically it's able to move from the quality control group to the assembly group to the annotation group. And it lets all of that kind of data processing part of the workload go through automatically. And then there's something called automatic QC. So there are these thresholds that can be triggered, and data that fails QC is flagged for review by the analysts. So there's a lot that we can do without manual intervention, and we're capturing a lot of metadata and information about what's being generated. And this has actually enabled us to use that Mongo database instance to provide a new kind of search across all of the kingdom groups within JGI. And so this has been a focus of ours for the last couple of years because if you're a user of the JGI and you've sent us DNA or RNA, you understand how to find data within our systems. We had things set up basically so that most of the identifiers were kind of internal identifiers that we understood. Like if I were to say sequencing project ID to this group, you might be able to extrapolate what that means and say, well, I bet there's a sequencing project and this is the identifier for it, but that's not necessarily something you would come to a search engine to look for, right? You might be wanting to search based on like an organism name or something else that's better understood. And so what we did is we took all of the metadata that was in JAMO, we took the metadata that was in the Kingdom portals, and there was a harmonization process that was done to actually align all of the more sort of, I don't know, human readable, I guess, non-JGI expert understandable fields. So taking things from a sequencing project ID and translating that instead to the organism name or to the type of study that was done or to the environment. And so now folks can come in and they can do a search that's based on these things that are a little bit more familiar. I'm not sure I'm explaining that terribly well, but it was actually really painful to go across all of our different groups and get people to broker agreements on how to search by these scientific names, where it was really easy because of our history to enable search based on like our internal identifiers that don't make it anything to the external community. Anyway, and this process has all been done with a lot of intentional user-centered design. So with each iteration of the data portal, we've gone and worked with our user community to find out what works for them and what doesn't. And something that's maybe potentially of interest is this file availability says now, but this is an indicator of whether or not the file is actively on the file system or if it's stored in tape. And one of the things that we do is that if you were to come to JGI and say, I would like all of your data, we're probably going to ask you, are you sure? Because that's a lot of information. But one of the things we would have to do is restore most of that from tape for you before you could download it, because it makes us, enables more efficient use of the more expensive resources. So like you can keep stuff in tape and then just restore it when people actually need to use it. And the kind of crazy thing about JGI that I didn't appreciate before working there is people do want the historic data. They want that data from 2013 or 2011 because the samples that have been used to generate these different genomes are so unique. And so we make crazier use of the tape archives than most other DOE users, which is kind of cool. And so another thing I just wanted to put in a little promo for a talk that's happening this week in case you happen to be going to the Code for America Summit. This is a talk that's more specifically about this effort to align metadata across all of the different kingdoms so that the plant group, the fungal group, the metagenomics group, the viral group are all using the same terminology to talk about their genomes and their organisms. And really some of the social engineering that was involved in making that work, because I think as one of the folks who asked a question earlier alluded to, a lot of this is driven by the need for culture change and the acknowledgement that folks have to work together, even when you're part of the same organization. So there's a link to the talk that's happening Thursday or tomorrow. And so this technology that we have is built on an Elasticsearch instance with a bunch of different indices across all of the metadata that's in JGI. So this includes our laboratory information management system data, which has information about all of the samples and the environmental data that was provided. There's the genome's online database, as well as all of the analysis that was done to those different objects. And then there are services that sit between the Elasticsearch instance and what a user would interact with. So the API that sits on top of this that anyone can access, including our partners at KBase. So just wanted to share a quick note about our computing infrastructure at JGI. So historically, our infrastructure has been at NERSC. In 2010, there was a partnership that formed between NERSC and JGI to shift JGI out of this mode of running everything themselves to a large-scale supercomputing facility. And that was really driven by the increase in data that I showed earlier. And so just the need for more computing power next to the data to let us get our work done and also to do some large-scale analyses. One of the exciting things is that Berkeley Lab is on a major fault line, and NERSC is right there where I've drawn the arrow. That's pretty close to this red fault. So I think everyone is probably pretty familiar with the fact that California has earthquakes. Some of them can be pretty devastating, and we're just waiting for a large earthquake to happen on the Hayward Fault. And there's a lot that's been done to the building to make it seismically sound. But one of the questions we got is, hey, most of your data or a lot of your data is really unique and irreplaceable. Somebody has gone out and tromped into the Arctic and taken a sample from some permafrost that maybe has melted by now. And so you really can't replace that sample or that data. So maybe you shouldn't keep it all on the fault line. And so we moved a bunch of this data to Oak Ridge for safekeeping. We did this migration of data over 5 days using Globus. So we were able to, basically not quite as straightforward as clicking a button, but pretty close, of just doing a bulk transfer from one organization to another. This is enabled by the DOE Energy Sciences Network, or ESNet, which gives us good connectivity between these different computing facilities. And this was also pretty cool because it enabled Oak Ridge scientists to have access to the data, but this would only be enabled if they also had access to the right metadata. And so one of the things that we've been wanting to do is actually make the JAMO metadata somehow accessible to folks at remote facilities. And as a result of having some of this data be more accessible, Gordon Bell Prize winning paper actually used all of this well-characterized publicly available data to look at the genetic underpinnings of opioid addiction. And this was something that used the entirety of the Summit system and was partially enabled by having JGI data hosted and findable within their computing environment, which is pretty cool. So access to large amounts of this omics data enables, even if it's about microbes and things you might not think are directly applicable to thinking about mechanisms for addiction, it turns out we're all made of the same chemicals and we all evolved from the same origin in the tree of life. And so there are relationships that you can find that go across the whole of the tree of life, and you need supercomputing power to be able to do that. And so when you think about or do kind of a reframe on what you're studying to really consider all of those different organisms, there's a whole lot of different hypotheses that people can come up with that they could explore with all of this data, which is pretty cool. But you also have to have the corresponding math skills and computational power available. So California not only has earthquakes, but we also have fires. And I swear I'm going somewhere with this. This is the IGB, the building I showed you at the start. That's where JGI does its operations. This is one of our friendly Berkeley Lab Hill turkeys. And I just think this is such a sinister landscape. And this is what it looked like September 9th of last year when we had some really bad fires and smoke and haze in the air. This is midday and it just looked otherworldly, like you're on Mars. And a lot of this sort of wildfire risk has actually led to PG&E, our electricity provider, doing public safety power shutdowns, which translates to the entirety of Berkeley Lab actually being without power. And so these things that are a little bit beyond our control really drive home the need for us to be able to distribute our data and analysis to maintain a high level of scientific productivity. So what steps can we take to actually make this possible? We've got a data management system, so that helps, but we also need to be able to distribute our compute. And so JGI has been working on a centralized workflow system called JAWS. The objective is being able to run the same pipeline in a number of different locations. And we really want everything to be portable, reusable, traceable, and the platform needs to work pretty well. So when somebody submits a job, they need to have some confidence that the job's going to execute and they'll get results back. And so right now we have this system up and running at NERSC and Berkeley Lab. And one of the challenges with distributed computing that this group is probably aware of is that you have to manage multiple user accounts. Different facilities have different policies. So one facility might use Slurm for scheduling, one might use UGE, but even if they're both using Slurm, they might have their queues configured in a totally different way. The available architectures could be completely different. One place might have CPUs, one might have GPUs, one might have, large memory nodes, one might have, just 24 gigs of memory on their nodes. And so you really need to be able to kind of tailor your submissions to these different facilities. When I step back and think about this in my role as the head of computing and informatics for JGI, where I don't want scientists spending their time is figuring out how to make their job run at a different facility, figuring out how to get a new user account, figuring out how to move, what's required to move data. And so if we can build a system that abstracts away the need to really think about where your job is running, kind of like what you can do in the cloud, you don't, that's hardware agnostic, then, we can let the scientists focus on the data that they're producing and the analysis and the hypotheses, and that aspect of the work can just be done for them. And also say that one of the other challenges is portability is a lot of work. Containers have made it easier, but they're certainly not a panacea for this. And so one of the approaches that we've taken is to use a workflow description language, or WDLs, to actually provide a specification for the pipeline as it's running. This is really similar to CWLs, or the Common Workflow Language specs that you can build, but this came out of the Broad Institute, which is another sequencing organization. And then we use Cromwell, which is a workflow management system that can ingest the WDLs and can ingest containers. And the Cromwell Workflow Manager then has backends that can submit jobs to these different computing resources. So the group that owns a centralized workflow system also configures Cromwell to be able to submit jobs where we have compute time, storage, and accounts. And so this provides a common interface to access all of these resources. And this is how we're providing access to cloud computing for JGI staff as well. And so what does this look like to a researcher? They come, they submit a job to our services that are currently running at NERSC. The data is, they do a lookup in JAMO to figure out what data is actually needed. Then this, the service authenticates using Globus at the different organizations to then transfer the data to the remote computing resource, and the work is executed and you get results out of that. And then once the work is done at this remote site, we actually bring it back to NERSC right now. So NERSC is kind of the home base for JGI's data. And that's because the bulk of what we have, our 10 petabytes, is sitting within the tape system, because that's the most affordable way for us to have that much data that we can manage. And so the data gets transferred back to the home repository with Globus, and then the data gets registered with the appropriate metadata with the JAMO system. And so this creates a bit of a closed loop where this work can be executed on any of those distributed resources that actually, where we have accounts and compute time. So application tokens are accepted by the facilities we're using to make it possible to transfer data on behalf of the user. So this is something that I thought was really cool that we worked with Rashna and the Globus team on, was basically making it possible so that the application, the JAWS application, is what's handling authentication. And so we can work with Lab IT, Berkeley Labs IT, with EMSL, with Oak Ridge, and say, okay, we're going to have this application user account, and this is the user that we actually want authenticating to initiate data transfers between NERSC and these other resources. And it made it a lot easier than asking a user to come in, use JAWS, and manage tokens on their behalf. So I thought this was a really nice setup that we eventually convinced all of the different security groups at these different resources that they could support. And so, yeah, just wanted to give a note about data movement between resources. We use Globus for that. We've been using Globus for a while. Back when I was at NERSC, I think this might have been the first time Rasha and I talked, was we broke it by trying to move tiny files that were all in the same directory, millions of them, because I had my little Globus pane pulled up and I was trying to move between two file systems within the same organization, and I just hit the go button, really naively transferring and moving data. So that was how we first got connected, and it's been fun to work with them ever since. And then JGI has a Globus endpoint that's set up for our users, and only the big users, these bioenergy research centers, which are pretty cool. They're DOE funded facilities that are doing research into biofuels. So a lot of cool data that they're generating, and a lot of need for generating sequence data to go along with it. So they use the Globus transfer service because they're generating a ton of data with us. But that some JGI users still just want to download everything through the browser. And I was looking through the data a couple of months ago, and I saw some people are willing to wait more than 9 days for a download to complete through the browser. And we reach out to them to say, hey, maybe you would like to learn how to use Globus. And so there's still kind of this tendency of people to use the tools that they're familiar with, even if something could be 10 times faster, which I find fascinating and gets back to this whole idea of user research and user-centered design. And then Globus is an integral part of our centralized workflow system. And I think without it, we'd have a much harder time moving data. So this is a summary. Hopefully you've learned that we produce a lot of complex, unique data for the scientific community. We're constantly deploying new instruments. Data is higher quality. Metadata can still be a challenge. We would definitely be lost without a good data management system. It was challenging at the time, and that's really enabled us to do a lot more with JGI's data and enabled the community to potentially do a lot more with that data. And then we're shifting to distributed computing for processing and large scale analyses out of the need for resilience to deal with fires, to deal with earthquakes, and also just to maintain a high level of availability and uptime for our users and our staff. And the data movement is faster and easier with Globus. So yes, that's my pitch for that. And then this is just a note on some upcoming calls and activities that JGI has going on. We have our own user meeting. Anyone's welcome to attend. And then if you're interested in actually going and collecting samples and having us sequence it, our community science program has a proposal deadline of July 31st. So, oh, and if you're a new investigator and you've never used us before, then there's a smaller call that you can apply to on September 15th. And with that, I will stop.
Thank you, Kjiersten. Fascinating talk. There's chatter on the team about how we see your passion for the science in this as well. So thank you. There are a couple of questions on chat, so I'll start with that. First one from Trevor Cooper says, do you control the applications using the tokens? I'd like to have the Globus timer application token, but I can't control the application's auth flow.
Yeah, we do. So we have complete control of the application and the environments it's running in. So that's a use case I hadn't considered before.
I guess it's also worth mentioning that they had to take the time to convince all the different sites to use the application method as well. So JGI pushed on that and got that. So the second question, this is from Bridget Raman. She asks about any effort to link the data in the JGI portal to data in other portals, or for JGI data to interoperate with other data portals, or is the JGI the single place where we have to get access to it?
That's a fun question. So I think one of the reasons we started the work, and I probably should have had some slides to explain this a little bit better for thinking about the metadata across the kingdoms, is because we want to be able to link our data to external repositories as well. And there's a huge amount of work that's happening with environmental ontologies that can provide more common semantic information. So you can take metadata that you've been capturing about an environment or a specific sample, and you can tag it with the appropriate ENVO identifiers, and then it gives you a common way to be linking to different repositories. And so that's pretty it's very early trying to do that. And so our first step was just getting our internal teams to agree on how to refer to organism names. And then the next step is to take our Genomes Online database, which has been some of this work is happening, to actually tie it to the environmental ontology, so that when we can start to do that. And then there's a lot of work happening within the National Microbiome Data Collaborative that will connect JGI data resources, particularly microbiome resources, with microbiome resources with NIH and NSF as well, or that's the vision for that. And the biggest pain point is getting the metadata to be consistent. It's fun, it is hard. I see Carl's question.
Yeah, you see Carl's question.
I'm not sure that I have. If I did, it would have been a few years back, because we were actually trying to move JGI off of an old file system onto a new one, and somehow I, as the consultant, wound up doing that data management, which was really what inspired us to build the JAMO system, because I was like, man, we can't do this again, and we're going to have to change file systems at some point. So I'm not sure. I could look back.
Actually,
We could
sorry,
try
others
it.
can't see the question, so I'm just going to repeat the question.
yes,
So
sure.
Carl Cornell's question is, Kjiersten, have you done any more million files in a directory transfer job since your first one? This is the question. So I haven't heard from her about anything failing, so either it worked.
Yeah, if we did, it worked.
Anybody else? Any other questions? So I have one. A lot of the discussion has been around providing the data to the user. So I'm hearing a lot of, so some analysis and providing data. Has there been thought or investment in value-added services? So rather than me download the data, could I be using the fantastic resources JGI has access to and doing more of my downstream analysis?
Yeah, so that's one thing that is provided through some of the science gateways that are organism specific. So you can go to Phytosome and you can do comparative analysis across plant reference genomes. And most of the tools that they've implemented there are really driven by the types of analysis that the user community wants to do. But again, those efforts are kind of very focused on a specific organism, and the groups who are doing things like the Gordon Bell work, where they want to look across all the organism types, they're organism agnostic. They're constructing these large graphs or networks that connect all of the genomes together and look for patterns and suggest different hypotheses. And those networks are something that are being put into K-base for other folks to explore. So there's something called the Department of Energy Systems Biology Knowledge Base, or K-base. And that's where I think, or where I'd like to see a lot of bespoke analyses happening that users or the community would like to do. So you can go into K-base, you can use the API to find JGI data, pull it into that resource, and then you can use all of the tools that they have available to do your analysis. Or what we've also done is we've got a partnership with NERSC to provide access to their computing resources for our user community. So we have an application that goes out roughly once a year, where we offer to work with people to say, hey, you want to do something crazy with a large amount of JGI data. Here's how we can help you do that on the NERSC resources. And we're trying to get our framing and our messaging around this, because I would say that JGIs really trying to catch up with where the data community is going and where our own scientific community is going. And, so we're trying to learn from them what would be most useful, but definitely these kind of science gateways and analysis platforms where you can either move a small computational analysis to the data or move small amounts of data to the compute just seems to be where everything is headed. And so our first step was to build the API to the data, and then hopefully we can build some cooler tools with the community.
Great. Okay, we're going to wrap up this talk here. Thank you very much, Kjiersten. That was a wonderful talk, and thanks for joining us.